The University of Washington 2005 Science Forum features faculty research on the impact of human advances on our environment and within populations throughout the world. Toby Bradshaw looks at the long history of engineering plants for us to eat and explodes myths in the controversy over genetically engineered food. Marine biologist Virginia Armbrust looks at how human activities are affecting microscopic phytoplankton, which contribute 50% of the planet's oxygen. Chris Diorio introduces us to the little radio transmitter chips that will soon replace barcodes in tracking potentially everything in our world. Steve Gloyd talks about healthcare systems in poor countries and the obstacles they pose to delivering the affordable and effective AIDS treatments that are now available. And King Holmes looks at the prevention of sexually transmitted infections and tries to unravel why people don't take precautions. Join us as we determine the effects of human imprints. Exploring Our World is made possible by the University of Washington Alumni Association, the College of Arts and Sciences, the Office of Research, the College of Engineering, the School of Medicine, the College of Ocean and Fisheries Sciences, and the School of Public Health and Community Medicine. Additional funding for this program is provided by UW Medicine and the University Bookstore.
Good evening. Welcome to the first lecture in the 2005 UW Science Forum series. I'm Ron Irving. I'm the Divisional Dean of Natural Sciences in the College of Arts and Sciences. I'm happy to be your host tonight. The generous corporate sponsors for this series are the University Bookstore and UW Medicine. This is the 6th year of the Science Forum series. It began as a partnership between the College of Arts and Sciences, of which both Toby Bradshaw and I are part, and the UW Alumni Association. It's since grown in popularity and in scope, and it's now put on by 5 different schools and colleges in the university: College of Arts and Sciences, the School of Medicine, the College of Ocean and Fishery Sciences, the School of Public Health and Community Medicine, College of Engineering, and also by the university's Office of Research. One of the goals of the series is to provide you with an overview of the very exciting research being done throughout campus. Partly, as you just heard with these 5 schools and colleges, science is being done in every corner of campus by people with a wide range of interests and backgrounds, and you'll see some of that tonight. We are, though it's not very well known, the leading public university in the country in federal government research funding. We get close to $1 billion a year in grants from the federal government, and that's testimony to how important the impact of our research is. Let me tell you a bit about our speaker. The speaker is Toby Bradshaw. Toby came to the university in 1984 as a postdoctoral fellow. In 1989, he joined the Department of Biochemistry, which is in the School of Medicine. He later moved to the College of Forest Resources, and just a few years ago moved to the College of Arts and Sciences as a faculty member in the Department of Botany, which through a merger 2 years ago became the Department of Biology. So Toby is now a professor in the Department of Biology. He's associate chair in the department for the graduate program, and just a couple months ago became the Washington Research Foundation Professor of Biological Science. Toby's interests are very broad. He is an evolutionary geneticist. Originally, his background and training were in molecular biology, but he had a lifelong interest in organismal biology. And now he uses a combination of laboratory, greenhouse, and field experiments to study adaptation in plants and animals. He works with ecologists, physiologists, evolutionary biologists. He's really a fantastic example, again, of the breadth of science done here, in this case, all in one person, across the different departments and colleges at the university. And he's also a wonderful teacher, a very popular teacher in the biology department. He really does it all and does it all well. And as a result, we're really in for a treat tonight as we hear about the science behind the controversy over genetically engineered foods. And if you get hungry during the talk, you can come up here and get some food. So please help me welcome Toby Bradshaw.
I'd like to thank Ron for that generous introduction. I'd like to thank you all for being here tonight. Ron described some of my research, which is in the area of evolutionary biology, evolutionary genetics. And even though part of my work does involve the genetic engineering of plants, it's a relatively minor part, and certainly I don't genetically engineer food plants. Nevertheless, I have some recent personal experience with the controversy over genetic engineering. 4 years ago, my lab and office at the UW Center for Urban Horticulture were firebombed by the Earth Liberation Front. You can see the fire glowing in my office here from 5 gallons of gasoline and a toy rocket motor. The Earth Liberation Front was concerned that I might be genetically engineering trees. I wasn't, although I wouldn't apologize for it if I was. The whole building that I worked in, Merrill Hall, was destroyed by this fire. If you look in what's left of my office, all you can see is a melted computer. I recovered a few keys and a doorknob. So it was pretty much a total loss for my lab in terms of infrastructure, but even worse than that, it destroyed the research and teaching collections of many of my colleagues at the center. Fortunately for us, the University of Washington is a very resilient organization. The state legislature was lobbied for funds. Private citizens who had built the center in the first place again donated their funds to rebuild the Center for Urban Horticulture, which just opened its new building a few weeks ago. So it's a pleasure for me to be here tonight to talk to you because I feel like this community has a real commitment to freedom of inquiry, freedom of scientific expression, and this controversy over genetic engineering is something that really deserves a lot of thought and attention. I hope that by the end of today's lecture, you are looking at your food in a completely different way. You should, by the time we're finished here tonight, know a little bit more and perhaps worry a little bit less about the genetically engineered food that we see in our food supply today. But on the flip side, you may know a little bit more and then worry a little bit more about the food that we consider to be sort of our ordinary food supply, and I have some examples of that up here today. Nearly everyone in the United States consumes genetically engineered food. Some people know it and some people don't. 3/4 of the soybeans that are produced in the United States are genetically engineered, and about half of the corn supply. And of course, these basic commodities are processed into all kinds of things. I've brought some examples of that. So we have soybeans that are processed into oil. We have corn that's processed into things like these chips. I'm not trying to push any particular brands here. These are just the things that I found at Larry's Market when I was shopping on Sunday. And then corn is processed into other things like artificial sweeteners or the high fructose corn syrup, not cane sugar that we're used to using for a sweetener, but high fructose corn syrup that's used in soft drinks. So nearly everyone has eaten or has drunk the products of genetic engineering if you live in the United States. The first genetically engineered food item that was made available in the United States in 1990, so this was 15 years ago, is cheese. You may or may not know, and you may not want to eat cheese after you do know, that the first step in making cheese is to mix milk with an enzyme that causes the milk to curdle, and then of course the cheese is processed from those curds. The source of the enzyme that curdles cheese traditionally is the lining of calf stomachs. So calves are killed, their stomachs are taken out, chopped into little pieces, dried, and those dried bits of calf stomach are thrown in with the milk in order to make it curdle. And that's how cheese is still made in Europe today. But in the United States, the gene that encodes this enzyme, chymosin, was cloned from cows. The gene was introduced into bacteria by genetic engineering, and now chymosin for making cheese comes from these genetically engineered bacteria. So it's a cow enzyme, but it's being produced by bacteria for the purpose of making cheese. So even if you didn't know it, you've probably consumed genetically engineered food. This is a great example of a genetically engineered food. Hope you can all see this. It's a papaya. Papayas from Hawaii are all genetically engineered to resist a virus, the papaya ring spot virus, which threatened to wipe out the entire papaya industry in Hawaii. By engineering the papaya to be resistant to this ring spot virus, the industry now is thriving in Hawaii, and this is the only example of a commercially released genetically engineered tree. We've been genetically modifying plants and animals for purposes of agriculture for about 10,000 years. We've only been genetically engineering plants for about the last 25 years. I'm going to make a distinction here between genetic engineering and genetic modification. Very often in the popular press, we see the products of genetic engineering described as genetically modified organisms, or GMOs. This is really an inappropriate terminology. The reason is there are all kinds of ways to genetically modify organisms that don't involve genetic engineering at all. We're going to talk about how some of those work. So I want to define genetic engineering as the transfer of genes from one organism to another by an asexual process. That is, it doesn't involve mating of animals, it doesn't involve pollination of plants. It's an asexual means of gene transfer. Genetic modification, on the other hand, takes in a broad array of technologies, many of which have been used without any conscious knowledge by early farmers. So I'm going to define genetic modification as change in the genes or genetic material by any means. So these could include things like mutation, they could include chromosome doubling, which can happen spontaneously or can be induced by chemicals. By selection, that selection could be artificial or it could be natural. Or by hybridization, that's by crossing two different individuals, sometimes two individuals that aren't very closely related. Lots of different ways to genetically modify organisms that don't involve gene transfer by asexual means, that don't involve genetic engineering. I want to give you a short outline of the talk. We're going to cover a few different topics. Why is it that plant genetic engineering is so controversial? Obviously, it is a very hot topic in society today. It's the source of trade disputes between the United States and the European Union. Lots of divided opinion within this country about its suitability for food production. Why do we genetically engineer plants? What's the purpose of genetically engineering plants? Why would anybody want to do this? And assuming that we do want to do it, how is it done? We have a 10,000-year history of plant genetic modification that predates genetic engineering. How was plant genetic modification done before genetic engineering was invented? And then the issue that concerns most people, does genetic engineering as a technology pose any unique risks, any risks with which we're unfamiliar after 10,000 years of agricultural history? Are there things about this process that should make us especially nervous? Why is plant genetic engineering so controversial? I asked my Biology 101 students, I polled them when I was teaching Biology 101. Every year I would poll my students, ask them, what is it about genetically engineering plants that makes you nervous? And I got the same answers every time, and I'm going to show them to you, and I bet they're the same things that are occurring to you as well. The first thing that always comes up is genetic engineering is unnatural because it breaches the species barrier. We think of species as distinct entities that don't interbreed. That's why they are distinct species, because they don't interbreed. Any kind of breakdown of that species barrier is inherently unnatural, and unnatural things are bad. Students are always concerned about the potential for risks to human health. They're equally concerned about potential risks to the environment from these genetically engineered organisms. And then this issue comes up every time as well, and that is, with genetic engineering, aren't we concentrating the power for food production in the hands of a few large corporations? Isn't that bad for food production in general, for food security, and especially food security for poor people? This is an issue that I won't spend much time on during the rest of the lecture, but it's something I'd be happy to talk about during the question and answer session. This part of it doesn't have so much to do with science as it does to do with public policy. This debate is incredibly polarizing. And we see evidence of that within our own society and between countries. But I think this is a pretty typical response. This comes from Prince Charles, who may not be a completely disinterested party here since he founded an organic food company, but let's assume that his motives are pure. He says, I happen to believe that this kind of genetic modification takes mankind into realms that belong to God and to God alone. And of course, scientists have responded with their characteristic humility. And Jim Watson, who won the Nobel Prize for describing the structure of DNA, says, in all honesty, if scientists don't play God, who will? That might be a bit presumptuous. The reason that this debate, I think, has been so polarized is because it's taken place largely in the absence of an appreciation of the history and science behind plant domestication and breeding that's given us the crop plants that we consume today as part of our food supply. So we're going to spend some time tonight talking about how plant breeding works, how plant domestication works, how plants have evolved under the natural forces of mutation and selection managed in an unnatural way by humans to produce from wild plants the food plants that we consume every day. Why do we genetically engineer plants? I'm going to argue that we genetically engineer plants for exactly the same reason that we've genetically modified them for the last 10,000 years, by conventional methods, by what are called conventional methods. We're interested in increasing the yield of crop plants. Most of the calories in our diet come from crops. These crops are all domesticated plants. Domestication is the first step to increasing yield. We need to adapt crops to new environments so that they can be grown in different parts of the globe. A lot of times, yield is diminished by attack from pests or pathogens. A lot of plant breeding effort is expended to try to breed in resistance to pests and pathogens. And then in the 1970s, there was a tremendous effort that led to the Green Revolution to produce short-statured cereal plants like wheat and rice that were capable of assimilating large amounts of nitrogen from fertilizer to produce very fat, heavy seed heads full of grain. And so there's been this emphasis on dwarfing genes for increasing yield in crops. It seems paradoxical that dwarfing a plant could make it increase in yield, but in fact that's the case. There's emphasis on improved quality and variety. If you've eaten tomatoes from grocery stores, you may not believe me when I say that plant breeders are actually interested in improving quality and variety, but that's been a goal of plant biology and plant breeding. Probably the best known proponent of variety is Luther Burbank, who hybridization experiments are famous around the world, and we still grow many of the cultivars that Luther Burbank produced in his breeding experiments. Breeders have always produced plants for profit, so food plants, we have production of improved seed by seed companies. We have plenty of examples from, say, the horticultural industry, not food plants. The famous tulip speculative boom in Holland is another good example of breeding for profit. The profit motive is still there in plant breeding today. The most important part about this technology for me is its use in basic research. Basic research to look at the plant form, function, plant development, the evolution of plants. This is a key area of plant biology. We can all remember Mendel's famous work with garden peas. He was using plants as an experimental system. Barbara McClintock, who won a Nobel Prize for her work on transposable elements, all derived from plant biology. And today, genetic engineering is just considered a state-of-the-art tool for discovering how plants work. Thousands and thousands of plant biologists around the country and around the world use this technique every day to genetically engineer plants to study the function of individual genes. I'm one of those scientists, and there are many of my colleagues around the country who do this. If you were to find out how ridiculously easy it is to make a transgenic plant or a genetically engineered plant, you might wonder why it isn't done even more often than it already is. So how is genetic engineering done? I found this on the Greenpeace website, and this is sort of typical of people who haven't kept up with their reading. On the Greenpeace website, it says that genetic engineering enables scientists to create plants, animals, and microorganisms by manipulating genes in a way that does not occur naturally. There's this emphasis on naturalness. Well, here's a natural example of genetic engineering. This large knobby growth on the tree is a crown gall. It's a large plant tumor. It's produced by infection of the plant by a small bacterium, agrobacterium. So on the upper left part of that, you can see the head of a pin, and then progressively magnified, you can see the size of the bacteria in relation to the size of the head of the pin. So these are very small organisms, 4,000 or 5,000 genes, but a very sophisticated system for injecting their DNA into the tissues of the plant, causing the plant to produce this tumor. The bacteria live inside this plant tumor. The details of this process of agrobacterium transformation of plant tissues, a lot of it was worked out right here at UW by Mary Dell Chilton, Milt Gordon, and Jean Nestor. Jean's in the audience today. Jean will be giving a lecture tomorrow in the Science Forum Colloquium that describes in molecular detail how this process takes place. But I want to give you just a short version so you can appreciate that The genetic engineering of plants is not at all a human invention, it's a natural process. Basically, bacteria are able to exchange DNA in a process called conjugation. This conjugation process vaguely resembles the normal sexual mating that we think of in organisms like animals and plants. And Agrobacterium has essentially adapted its conjugation system to conjugate with plant cells instead of conjugating with other bacteria. So it's sort of a very promiscuous mating system that Agrobacterium has evolved. Agrobacterium shoots its own genes into the plant cell. Those genes are taken into the plant cell nucleus. The genes are incorporated into the plant's own chromosomes and then transmitted faithfully generation after generation along with all of the plant's other genes. I was attracted to Milt Gordon and Gene Nestor's labs 21 years ago to work on genes that had been incorporated from Agrobacterium into wild tobacco plants. So you can go out and collect wild tobacco plants from nature and find the scars of past Agrobacterium infection in the genome of wild tobacco. So this is very much a natural process, but of course it can be used in ways that might be considered unnatural by manipulating the genes that are inserted into the plant, and that's what genetic engineering is all about. It's the unnatural manipulation of a natural process. Here's a great example of plant genetic engineering. It comes as a surprise to most people that this is a natural process, but it can be manipulated in interesting ways by plant scientists. This is a tobacco plant, but it's glowing green because it's been genetically engineered with a gene from a firefly, the luciferase gene. So this plant is glowing the same way that a firefly would glow, and for the same reason. It contains the gene from the firefly that makes the firefly glow. Now, to some people, this is a real monstrosity, a real aberration. But I have to say to most biologists, this is a beautiful proof of the common origin of all life. This shows that genes taken from a firefly, from an insect, when put into a plant, are expressed perfectly. They're expressed in just the way that they would be expressed in the firefly. It's a beautiful confirmation of the prediction that Darwin made 150 years ago that all life descended from a common ancestor. All of these organisms have the same genetic code as we discovered about 40 years ago. They can all interpret each other's genes and express them correctly. It's really quite a wonderful culmination of 150 years of scientific thought. So if we could only genetically engineer for the last 25 years, how are we genetically modifying plants in the 10,000 years that preceded the advent of genetic engineering by humans? First of all, the early farmers had the ability to perform artificial selection of spontaneous mutations, that is, mutations that just arise in natural populations by natural processes. A cosmic ray hits the DNA molecule, changes the DNA sequence, and changes the protein that's produced from that sequence, and somehow changes the morphology of the plant. Or spontaneous hybrids, crosses between plants that might rarely occur in nature, but when they do occur, they have some special properties. You can imagine that our ancestors, the ancestral farmers, were keen observers of nature. When they started harvesting grasses as food, they probably noticed that when they brought home some of these grasses to use as food, that some of the grasses still retain the seeds on the tip of the shoot. And of course, the plants that retain the seeds on the tip of their shoot were planted the next year for seed. A natural, free-living, wild grass plant never retains its seeds on the tip of the shoot. By dispersing its seeds at the end of the growing season, that's how the plant propagates itself for the next year. But a human harvesting these plants preferentially harvests mutations that cause the grain to stay on the tip of the shoot. And so by this artificial, sometimes unintentional selection, many of our cereal crops were produced, and you've all seen wheat fields in eastern Washington, I'm sure, with this beautifully erect stalk and this beautiful seed head containing all of the wheat seeds, and yet those seeds never fall to the ground. They remain stuck to the tip of the shoot, ready for human harvest. Such a plant can't exist in the wild. It can't disperse its own seeds. So it's a very unnatural mutation that has a fitness of 0 in nature, but which has very high fitness when it's managed by humans. Wheat is also another great example of spontaneous hybridization. The wheat that we use to make bread is actually a hybrid of 3 different grass species from 2 different genera of grasses, very unrelated species of grasses. There is no such thing as wild bread wheat anywhere in the world. It doesn't grow as a wild plant. It never has. It can't survive as a wild plant, and that's typical of all of our domesticated crops. 300 or 400 years ago, we began to accelerate this process of breeding and selection by, instead of waiting for spontaneous mutations or spontaneous hybridization, we started hybridizing plants deliberately, sometimes very wide crosses between very distantly related plants, crosses that would never happen in nature. So I brought along some examples of some of these wide crosses that have been done, and it may surprise you. So I have quite a variety of plants here. Here's one, an eggplant. What could you cross an eggplant to that might produce a hybrid? Well, here's what Luther Burbank crossed it to, tomato. So tomato and eggplant are not particularly closely related, but they can be hybridized artificially. Burbank also did another cross between the tomato and a potato. So tomatoes and potatoes, we never find those growing on the same plant. It would be sort of handy if they did, but they don't. But this cross can be made. It never occurs in nature, but it can be forced by humans to work. So even 100 years ago, these kinds of crosses were done all the time, sort of an exploration of the plasticity of plant genomes tested by hybridization. And then after World War II, we were able to bring even more powerful methods to bear on the process of generating genetic variation that could then be selected for in order to produce food crops. So I brought along an example of a plant that was produced by deliberate radiation using thermal neutrons from a nuclear reactor. So this is the Rio Red grapefruit. It was produced at Texas A&M University by irradiating the buds of a pink grapefruit. So pink grapefruit was mutated into red grapefruit. All of you who have enjoyed a red grapefruit have enjoyed the products of nuclear fission. As an interesting side note, in this country, organic food may not be irradiated before it's sold, but yet organic producers all the time grow plant varieties that themselves were produced by irradiation of the seed. More than 2,000 varieties of plants that are produced commercially now were produced by deliberate radiation or chemical mutagenesis of plants, and 80% of all the wheat varieties grown around the world were produced in this way. So again, it's management of a natural process mutation by an unnatural manipulation, radiation from a nuclear reactor. So if you thought that your plants were some kind of a pristine, wild plant, that if you were just in the right part of the world, you could walk outside and find your red grapefruit growing somewhere, I hope you're getting the idea that these plants are sort of grotesque caricatures of a wild plant. And I'm going to show you some examples of that here in a minute. So here's an example of genetic modification with which I'm sure you're all familiar. This is a wolf. It's the product of natural selection. Wolves have a very wide geographic range. They look about the same everywhere they live. Humans have genetically modified these wolves to produce dogs. A dog is not a different species from a wolf. It is a wolf, but it's a genetically modified wolf. It wasn't done by genetic engineering. It was done by artificial selection of mutations. So mutations arose in wolves that produced long hair or short hair or no hair. And we have dogs with all of those traits. We have dogs that point. We have dogs that trail foxes. We have dogs that herd sheep. Those are all produced, all those 153 breeds of dogs recognized by the American Kennel Club are all produced by mutation and artificial selection, a human manipulation of a natural process. Darwin used examples of domestication of both plants and animals as evidence that his theory of evolution by natural selection could occur. He saw artificial selection as a very strong example of selection. He expected natural selection to be much weaker. Humans can impose phenomenally high rates of selection because we select for all kinds of things that would never be able to survive in the wild. I mean, I want you to try to imagine that Chihuahua trying to survive somewhere in the wild. Let me tell you that a crop plant has no more chance of surviving in the wild than that Chihuahua does. So I've brought along some examples of wild plants, and I'm going to ask you to think about what the domesticated version looks like. So here is the wild plant, here is the wolf. I want you to think about what the Chihuahua is. It's a plant that you're all familiar with, that you all have eaten, I'm sure. So think about it, and I'm going to ask you in a minute, I'm going to hold up the Chihuahua for you to look at, and then I'm going to ask how many of you got the right answer. So, no cheating here. Okay, so let's look at this little red fruited thing. You can see how big it is. This is the Chihuahua. Right? This is a giant, mutated version of that wild plant. It's a different color, it's green. Instead of being blazingly fiery hot like that wild chili is, this is a very mild bell pepper. And of course, by mutation and selection, we've produced all kinds of other really cool pepper plants. Different morphologies. I have to be a little careful with this habanero here because if I get this on my fingers, it'll burn my eyes the next time I touch them. So we can produce all these different varieties. These are like little breeds of dogs. This is like the sporting dog group, a little spicy. Okay, let's look at another wolf. Now, I know there are a lot of good botanists in the audience, so I don't want you telling your neighbor what this is. And I've taken the title off the bottom. Think about what this looks like, and let's think about what the Chihuahua might be. So take a good look at this plant. Think about what you've grown in your garden, what you've seen at the grocery store. What is this going to be? Well, in this case, this plant doesn't just produce a Chihuahua. This thing produces the whole dog show. This plant is a wild cabbage, which, here's the tame cabbage. We've all eaten that. But there's more. Cabbage and kale are the same thing. They're the same species of plant. There's been different selection for mutations that produce variation in their leaf morphology. This produces this round, tightly constructed head. This produces these loose leaves. Are there any other things in the dog show here? Bok choy. Bok choy is the same species as cabbage, which is the same species as kale, which is the same species as cauliflower, which is the same species as kohlrabi, which is the same species as broccoli. And I couldn't find any Brussels sprouts because apparently no one likes those, and so they don't even sell those at Larry's anymore. But Brussels sprouts are also the same species. So these things are all the same species. You don't go out on the bluffs in England where this picture of this wild cabbage was taken. You don't go out and find cabbage and kohlrabi and Brussels sprouts and cauliflower. Those are all unnatural mutations that have been selected by humans because we like to eat them, or at least there's a few of those things that we might like to eat. They weren't the hottest items at Larry's, I have to say. There wasn't a long line in front of the kohlrabi stands. So now we're going to make this a little bit harder. Here's a wolf. What does the Chihuahua look like? It's a pretty small plant. It looks like it might be some kind of a grain. You can see some seeds along the central axis there, the rachis. This is one of the most remarkable transformations in all of biology. This is the Chihuahua. So this is corn. And if you look at the morphology of corn, with its many rows of seeds, with its cob, with its seeds enclosed entirely in a husk, which prevents it from dispersing its own seeds, this is an amazing transformation. There is no such thing as wild corn. Teosinte, the plant that's shown on the slide, is the closest thing that there is to wild corn. But it's completely inedible. Every seed is enclosed by a glume, a coating that's so hard that it will break your teeth if you try to eat it. But we've domesticated this. It was domesticated 7,000 years ago in the Central Plateau of Mexico and in Guatemala. But how this transformation took place is only now becoming clear. It turns out to be due to major mutations with huge effects, as you can imagine, given the degree of morphological transformation that we see. This is probably the hardest one that I've ever showed to my biology class. They have the most trouble with this one. So here's the wolf. I have to give you a little sense of scale here. This is about the size of your thumb, and those round things in the middle are seeds. They're about the size of a garden pea. So they're hard, black, round seeds. So that's the wolf. What is the chihuahua? What is the domesticated version of this? It's this. Yes. So this is a banana, and you're going, hey, I've eaten thousands of bananas. I've never found a seed in my banana. That's right, and you never will. The cultivated banana is not a wild plant. It can't be a wild plant. It doesn't even produce seeds. Why produce a fruit, which is meant for dispersing seeds? Why produce that if you don't have any seeds? The reason bananas, cultivated bananas, don't have any seeds is because they have 3 sets of chromosomes instead of 2. So some bright person walking through the woods say, hey, look, here's a banana with no seeds. This is good. They didn't know that it had 3 sets of chromosomes instead of 2, but it does. They did know that they couldn't propagate it by seeds because it doesn't have any seeds. Those little black specks in the middle of your banana are aborted seedlings. They can't make normal seedlings because there's no good way to divide 3 sets of chromosomes evenly to produce a sperm and an egg. So these are triploid, they're sterile, but they are tasty. So we eat them, but they're not anything like a natural plant. A wild banana is a real fruit with real seeds, but we never eat those. So I don't want you to think of crop plants that we've been producing for the last 10,000 years as natural. They're not natural. They're an abomination. They're a grotesque caricature of a wild plant. But they can be really good to eat, but they're not natural. They're not any more natural than the Grand Coulee Dam. They're not any more natural than a nuclear power plant. Every time you drive by a farm field on the landscape, you should be shocked. You should say, that doesn't look right. That looks out of place. Why is it that we think of plants that are used for food as natural? Because we have 10,000 years of familiarity with them. And so this argument about whether genetic engineering is natural is a bit of a red herring. There's no such thing as a natural crop plant, at least that's grown on any sort of large agricultural scale. Does that mean that agriculture is bad, or for that matter, does it mean that electrical generation by dams or by nuclear power plants is bad? We've harnessed, in the case of both electrical power generation and in the case of plant breeding, we've harnessed these natural processes. So in the case of dams, we've harnessed the hydrological cycle that pumps water up to the top of mountains for us. And we've harnessed the gravity that drops the water through the turbines below the dam to generate power. We've harnessed nuclear fission, something that happens in the middle of the earth, but something that we have to arrange to have happen on a big scale on the surface of the earth to generate steam, to turn turbines, to make electricity. These are natural processes, all natural processes, but they're all manipulated by humans in unnatural ways to produce things that have generally been perceived as good things. In the same way, plant breeding, we use things like mutation and hybridization and genetic engineering. All of those are natural processes too, but they're manipulated in unnatural ways by humans in order to produce food in a concentrated form. I'm going to argue that the concentrated production of both food and energy are essential to the modern society that we know. Even though we hold natural ecosystems in high regard, and we should, the systems that we use to produce food and energy are in no sense of the word natural. Both electrical power generation and agriculture have health risks and they have environmental risks. We need to be aware of those. We need to try to deal with them, but we can't pretend that they don't exist or that any of this is natural, and somehow if we made it natural enough, all the problems would go away. It's just not the case. I want you to think of a natural setting. If we went into the North Woods and saw a pack of chihuahuas chasing moose, That's about what an agricultural field is like. That same mental image should come to you every time you see an agricultural field. It should be like chihuahuas running through the North Woods. Just not going to happen. So the question really isn't whether plant genetic engineering has risk. I'll save you the suspense and tell you that like any technology, plant genetic engineering does have risks. So does every technology, including ordinary agriculture, the kind we've been doing for the last 10,000 years. It has some benefits, though. It gives us predictable food supply in the same way that hydropower and nuclear power plants can give us a predictable energy supply. There's some value in that. But there are downsides. There are risks. We know all about those here in the Pacific Northwest because you have great examples in the case of electrical power generation of the negative environmental effects of both dams and nuclear power. But we need to get electricity from somewhere if we're going to maintain society the way that it's structured today. So let's look at some of the issues that my students have brought up and see how much genetic engineering really differs from the kind of plant genetic modification that we've been doing for the last 10,000 years. We want to know, does plant genetic engineering pose any unique risks, any risks with which we're unfamiliar after 10,000 years of genetically modifying plants by conventional means? We have a lot of experience genetically modifying plants, 10,000 years worth. Is any of this applicable to the problems that have arisen as a result of genetic engineering? My students worry about genetic engineering being unique and breaching this species barrier. This is a bit of an unfounded concern. So I had to ask someone what this plant was. Not something I eat on a regular basis, and they only had 3 or 4 of them at the store, so I got the feeling that not too many other people were fighting for this one either. This is a rutabaga. A rutabaga is a cross between a turnip, which it sort of resembles, and a cabbage. So this is an unnatural hybrid. It has lots of extra sets of chromosomes because we cross 2 things that aren't very closely related to each other. There's a different example of exactly the same cross. This is oil from canola. So canola, when we grow this plant for its seeds and its oil, we call it canola. When we grow it for its root, we call it rutabaga. We don't have canola and rutabaga on the same plant, but they're derived from the same cross, an unnatural hybrid between turnips and cabbages. This is one of my very, very favorite weird hybrids here. So I don't have the plant, but I have some flour made from this plant. This plant is Triticale. Triticale is a cross not even between 2 closely related species. This is a cross between 2 different genera, 2 very unrelated groups of plants. And the name Triticale comes from the 2 genera from which this thing is constructed, Triticum, which is wheat, and Secale, which is rye. So Triticale is a wheat-rye hybrid. Wheat itself is sort of an unnatural hybrid, so Triticale is about as unnatural as it gets. The interesting thing about Triticale is that you may say, well, these are just 2 different grasses. Okay, so somebody put them in 2 different genera, wheat and rye. They look about the same to me. When you look at the DNA of wheat and rye are as different from each other as humans and chimpanzees are. So, when you look at this bag of Triticale, let's think about that as a really wide cross, as different as a cross between a human and a chimpanzee would be. That's how unnatural this is. I found it in the natural food section of Larry's, by the way. Plant breeders tried for 100 years to produce this hybrid because everyone understood that the high yield of wheat and the high environmental tolerance of rye would be a good combination for agriculture. For 100 years, there was failure until finally someone figured out how to overcome the problem. The problem with this hybrid is it's sterile. It has an unusual, odd-numbered set of chromosomes. By doubling that number, the plant suddenly became fertile. It was doubled with a plant compound called colchicine. So this chromosomes were artificially doubled by chemical treatment. And now Triticale has grown over a pretty large area around the world because it's very high in protein, and it's used to make things like this cereal that I just brought in. This is as unnatural as food gets, I think. Here's one of my favorite crosses between 2 different species. So I know you'd all like to believe that the strawberries that you can buy in the store now are some kind of a natural food, but I hate to break it to you, this is a hybrid between two species, one of which is found in North America and one of which is found in South America. We know to the exact year when the first strawberry like this was produced. It was in the early 1700s. And since then, it's dominated worldwide strawberry production. So even though this is completely unnatural, a weird hybrid is pretty good. And if you're interested in horticulture at all, so if we get away from food for a minute, the horticultural industry is full of examples of successful commercial hybrids. Roses, rhododendrons, orchids, these things are all hybridized, artificial hybrids. It's really quite remarkable how little people worry about hybridization, even though we're crossing things that are very, very different from each other, compared to the level of concern that we give over genetic engineering, for example. Many, many other crops are routinely crossed to wild relatives of different species, so there's a short list here. My students are always worried about plant toxicity. Oh, I'm worried if we genetically engineer plants, they're going to produce toxins. So the interesting thing about this is plants already do this. We have a lot of experience with it. This is a particularly nasty example. This is a blister. The blister on this poor chap's finger is caused by exposure to giant hogweed. Giant hogweed produces toxins that don't have their effect until the toxin exposed area of the skin is put in the sunlight. It's a phototoxin called psoralen. So when these compounds are stuck on your skin and you expose that skin to sun, these horrible blisters occur. Hey, well, what is that? I don't eat giant hogweed. Why is that a problem? Because giant hogweed has this relative that you probably do eat. Those nasty compounds are in these plants to prevent insects from eating them. It's a good reason for a plant to produce these poisons. So a lot of times when we have a cultivated variety where we've bred the poisons out so that we can eat them, we lose our insect resistance. To get the insect resistance back, we cross this to a wild relative. There's a good example from celery breeding where celery was crossed to a wild relative. It passed all of its agricultural tests with flying colors. When the first fields were planted, all of the field workers who came back in for picking the celery that day were covered head to toe with these blisters because someone had forgotten to check whether or not this phototoxin was being produced. There's no regulation, by the way. If you're a plant breeder, you can cross these things to anything, any other wild species, and put them on the market without ever testing them for any kind of safety. So plants produce nasty compounds. So let's look and see what plants are trying to do to us. We've already talked about chili peppers. Chili peppers are hot because they're trying to prevent mammal predators from eating them. And birds can't taste the heat in chilies, so birds are the preferred disperser by the chili because birds don't chew up the seeds. So the heat in chili peppers is really meant to deter us from feeding on them. It's a bit of a chemical warfare. We sort of enjoy that part of the warfare, but it gets worse after this. Most people think of potatoes as a pretty benign plant. Potatoes are actually a member of the deadly nightshade family. They're quite toxic. I brought along a friendly potato. I showed this one before. This is a nice potato. This potato here, which came from my own pantry instead of from Larry's, this little guy could make you really sick. Potatoes, all of the above ground parts of the potato, the stem, the leaves, are quite toxic. They're full of a compound called solanine that will cause you to become nauseous, to give you diarrhea, to get dehydrated, and if you eat enough of it, which isn't an incredible amount, you can die from it. Potatoes produce this in all the green parts of the plant. So when the potatoes in your cupboard start to grow like this, the skin becomes green, and these little roots that are coming off here are loaded with solanine. So if you have potatoes like this, don't eat them. They will make you sick. Oilseed rape, a pleasant name for a plant. Oilseed rape was used as a cooking oil until about the 1940s when it was discovered that it contained a compound called erucic acid. Erucic acid causes heart disease in people. And so the oil was only used as an industrial lubricant until plant breeders could breed the levels of erucic acid down low enough, below 2% of the total oil content, to make it safe for human consumption. So this rapeseed oil that was produced by breeders, this low erucic acid rapeseed oil, was approved for human consumption in 1987 by the FDA, so it could be consumed in the U.S. I guess the Canadians thought that rapeseed was probably not going to sell a lot of oil to consumers, and so they renamed it canola. So canola is actually the product of plant breeding to try to eliminate some of these poisons from rapeseed oil. Cassava is a staple of many poor people in third world countries. It's a root crop. The skin of cassava is loaded with cyanide. So if you eat unprocessed cassava, you will die. If you heat it and boil it, you can get rid of the cyanide, and then you can buy this, which is cassava flour for making tapioca pudding. So tapioca is derived from an incredibly poisonous plant, and cassava still kills people who aren't aware of how to prepare it today. It turns out that people who grow cassava prefer to grow the high cyanide variety of cassava because yields are higher. Insects eat less of this poisonous cassava than they do of the other kind. You may have heard of this bean, castor bean. You may have been, unfortunately, at some time in your life exposed to castor oil. After the oil is pressed from the seed, one of the seed proteins is a protein called ricin. Ricin is so toxic that 500 micrograms of it, 1/1000th the size of an aspirin tablet, is enough to kill a human, and it's even been used by various government agencies to assassinate other people. You may remember a famous case of a Hungarian being stabbed with an umbrella that had a little ricin pellet at the tip and killed the unfortunate Hungarian. So plants produce a lot of nasty compounds. In case you hadn't gathered by now, plants are not interested in being eaten. They're interested in propagating themselves. This will be bad news for coffee drinkers. So plants produce all kinds of other compounds, some of which are carcinogenic, lots of different compounds in coffee. 28 of them have been tested for carcinogenicity. Of those, 2/3 cause cancer in rats and mice. Luckily, I'm not a coffee drinker. I have all kinds of other vices, but I'm not going to die of cancer from drinking coffee. Plants produce lots of natural pesticides. We've already talked about some of those, but some of these are secondary compounds with really nasty structures. Many of them, more than half of those tested, cause cancer in laboratory animals. Pyrethrum is one of those. Pyrethrum is used on organic farms all over America and all over the world. It's carcinogenic. It's as carcinogenic as the synthetic pesticides that people seem to worry about so much. Is genetic engineering unique in potentially introducing allergens? There's a story in genetic engineering of this experiment where Brazil nut protein gene was introduced into soybeans in order to improve the nutritional quality of the soybean. This had to be abandoned during development because it was quickly realized that people who ate the soybean might be exposed to this Brazil nut protein to which they were allergic. They would have no way of knowing when buying a soy-based product that it might have this protein in it. But we're quite familiar with allergens in traditional agriculture as well. About 3 million Americans are allergic to peanuts, and it kills about 100 Americans every year. So this is a plant that I don't hear anyone calling to remove peanut butter from the shelves. Wheat gluten, another 3 million Americans are allergic to this. It causes celiac disease, malabsorption of nutrients. But these are things that we don't seem to care about so much because we're familiar with them. Are there unique environmental risks that genetic engineering poses? We've had herbicide-resistant crops. You're probably all familiar with the Roundup Ready version produced by Monsanto, but we've had herbicide-resistant crops produced by ordinary breeding for the last 50 years. Whether they're produced by genetic engineering or by conventional breeding, they have exactly the same benefits. That is, that we can use no-till agriculture, which is a great soil conservation measure, but they have the same risks as well. They can lead to the evolution of resistant weeds. They can lead to increased dependence on this chemical weeding, which if you're a chemical company, this can sound like a good thing, but to others of us, it doesn't sound so good. But they're not, the risks aren't unique to genetically engineered food. What about environmental risks unique to genetically engineered plants? We have insect-resistant plants that have been produced by both genetic engineering and by conventional breeding. But regardless of how they're produced, they have the same benefits and they have the same risks. It's not the process that matters, it's the properties of the product that matter. I want you to remember this. Not everything that is natural is good for you. I've given lots of examples of that. Not everything that's good for you is natural. Vaccines are not natural, but we all use them to prevent disease. We have 10,000 years of experience with many of the exact same risks that are posited for genetic engineering. So we have a lot of familiarity with these. We should use that information. And again, I want to reiterate, it's the product, not the process, that matters. This is a science-based definition of safety. It's product, it's not process. Are there any products that might be of unique concern? I would worry about transgenes that encode common allergens. The most common allergens are to wheat, to peanuts, to shellfish, and to eggs. We probably don't want those engineered into our food supply and staple food crops. Pharmaceutical proteins are particularly problematic because these are by definition active at very low concentrations. Having those in a food supply that can't be well segregated is probably a dangerous thing. Are there products of plant genetic engineering that might increase public acceptance of this technology, to which there seems to be so much resistance? I think if we could improve the nutrient content and flavor, lots of us could get interested in this technology. There's a development of edible vaccines, especially for poor countries, especially for childhood diseases, because these are much more easily transported than typical vaccines. Could we engineer non-food plants to produce industrial raw materials to replace petroleum? There's a lot of interest in this. And could we do something that companies will never do, and that is genetically engineer crops for low input, sustainable agriculture, something that a company that's interested in high inputs can't do and won't do. Let's just, in closing, put genetic engineering in perspective. In the United States, we have converted 300 million acres of our ground to grow the 4 major crops that we grow in this country. That's the area of Washington, Oregon, and California combined. That is a large-scale environmental impact. When we introduced those 4 crops into this country, none of them are native. They brought with them 200,000 genes that had never been tested in the United States. Genetic engineering has introduced about a dozen new genes. They have all been tested. What is the scale of worry that we should apply to these various technologies? And I want to say something about this because it always comes up. Doesn't it bother you to put flounder genes in a tomato? So flounder genes have been engineered in tomatoes to try to make them more cold tolerant. I want to tell you that the reason biologists don't worry about this is because tomatoes already have flounder genes in them. Wild tomatoes have flounder genes in them because all life is descended from a single common ancestor. The red areas here show the protein sequences that are identical between human, flounder, and potato because I couldn't find tomatoes. A quote from Charles Darwin telling us that ignorance produces confidence, but it should not. Science is the best system ever devised for dealing with uncertainty. We need to use science to deal with that uncertainty. And I want to tell you that no matter what your mother said, you are not what you eat, you are what you know. Thank you all.